Metabolism of protein. The functions of proteins are bodybuilding. They helps to build the body. So during metabolism, the protein are digested into amino acids were in the digestive tract and absorbed into the blood and enters the liver. So liver is a storehouse. It also seen in the blood. So the amino acid is digested, then it is absorbed into the blood and then move to the liver, blood to the liver. So the blood amino acid comes from the absorption from the intestine, synthesis. The body can be synthesized the amino acid and by the hydrolysis of protein, the broken down protein. So this is the way by which the amino acid in the blood is increased from this amino acid pool. So this is called the amino acid pools. So what is an amino acid pools? So all the amino acid that is seen in the blood is called amino acid pools. Where it comes through absorption, synthesis and protein hydrolysis. So the excess amino acids are broken down into ammonia. If there is an excess amino acid, if there is lot of protein intake, then the excess amino acids are broken down into ammonia and other nitrogenous products. And what is the use of these amino acids? These amino acids are taken by the tissue for its various metabolic needs. So the amino acids are used in the following ways. So first one is called deamination, transamination, decarboxylation and transmethylation. So these are the four steps that is involved in the catabolism of amino acid. Let's look each one. So the first one is deamination. It takes place in liver. It means the removal of amino groups from amino acid, and what happens? Produce a ammonia and a keto acids. And there are two steps in this process. So one is the oxidation or removal of hydrogen and hydrolysis adding water. So let's look at the first steps. So this is a sample amino acid. So where two hydrogen atoms are removed, the process is called oxidations. This one. Then what happens if two hydrogen atoms are removed, one from here and one from here, and what happens? C double bond NH is created. This is called amino acids, where water molecule is added and broken down and ammonia is produced. Ammonia is produced. It creates where a C double bond is created here. So this is called keto acid. Removal of amino group from the amino acids usually occurs when nitrogen is rich in diet. കൂടുതൽ nitrogen protein diet ഭക്ഷണത്തിലേക്ക് വന്നു കഴിഞ്ഞാൽ oxidative deamination നടക്കുക. It splits amino acid into ammonia and keto acid created. It is catalyzed by the enzyme amino acid dehydrogenase. So these hydrogen atoms are removed by amino acid dehydrogenase enzyme. Let's look one examples. So one of the examples is glutamic acid. So this is the structure of glutamic acid, and the first step is what? Oxidation, where the glutamic acid dehydrogenase remove two hydrogen from here. It removes two hydrogen from here, and what happens? It becomes, so the two hydrogen atoms are removed from here, and that is accepted by NAD here. The NAD is accepted the hydrogen atoms and it becomes NADH. So the glutamic acid becomes alpha aminoglutaric acid. So this is the first step, removal of hydrogen atom, and the second step is from the alpha aminoglutaric acid, from here water is added and hydrolysis takes place, where it becomes the nitrogen atoms NH is removed here and ammonia is produced. So now the alpha aminoglutaric acid become alpha glutaric acid. Alpha glutaric acid is produced. So this is oxidative deamination. Very simple steps. In the first step, 2 hydrogen atoms removed, and the next steps, water is added and ammonia is removed. So new amino acid are created here. So the glutamic acid is converted to a new amino acid that is called alpha glutaric acid. Next process is transamination, other protein catabolism. So this is transfer of amino group from an amino acid to an alpha keto acid. It is received by alpha keto acid, by which results in the formation of new amino acid and a new keto acid. So this is an examples. So this is an alpha amino acid. This is another keto acid. So this amino group is transferred to here. So this become keto analog of alpha. This become this, and this become this one. Okay, a new amino acid created. The significance is that it brings up about the interconversion of amino acid in the metabolic process. സ്വല്പം നമ്മുടെ ശരീരത്തിന് ആവശ്യമായ എല്ലാ അമിനോ ആസിഡും നമ്മൾ ഭക്ഷണത്തിലൂടെ കിട്ടണമെന്നില്ല. അപ്പോൾ കിട്ടിയതിനെ കൊണ്ട് മറ്റൊരു അമിനോ ആസിഡിനെ ഈ ഒരു പ്രോസസ്സ് വഴി ട്രാൻസാമിനേഷൻ വഴി ഉണ്ടാക്കാൻ വേണ്ടി പറ്റും. The process is characterized by the enzyme transaminase or aminotransferase and in the presence of coenzyme pyridoxal phosphate. Okay, the coenzyme receives the amino group, pyridoxal phosphate receives the amino group, forming pyridoxamine phosphate, which in turn transfer the amino group to the keto acid. എവിടുന്ന് ഈ അമിനോ ഗ്രൂപ്പിനെ ആരിലേക്ക് ട്രാൻസ്ഫർ ചെയ്യും? നേരത്തെ പറഞ്ഞ pyridoxal ഗ്രൂപ്പിലേക്ക് ട്രാൻസ്ഫർ ചെയ്യും. ആ pyridoxal ഗ്രൂപ്പാണ് ഇതിലേക്ക് ഇവിടത്തേക്ക് ട്രാൻസ്ഫർ ചെയ്യാൻ വേണ്ടി സഹായിക്കുന്നത്, ഒരു ട്രാൻസ്ഫർ മോളിക്യൂളായിട്ട്. So the transfer of amino group from, so this is one of the examples. So this is the alanine amino acid where the amino group NHD group is transferred to alpha ketoglutaric acid. This portionലേക്ക് ട്രാൻസ്ഫർ ചെയ്യും. അപ്പോൾ ഇവിടുന്ന് ഇത് ട്രാൻസ്ഫർ ചെയ്യുന്ന സമയത്ത് NH3 ട്രാൻസ്ഫർ ചെയ്യുന്ന സമയത്ത് ഇവിടെ ഒരു C double bond O ക്രിയേറ്റ് ചെയ്യും, this one, and this is called pyruvic acid. So what happens? The alanine becomes pyruvic acid and the alpha ketoglutaric acid becomes, by receiving an amino group, it becomes glutamic acid converted. Clear? So അങ്ങനെ ആൽഫ കീറ്റോഗ്ലൂട്ടാറിക് ആസിഡിനെ ഗ്ലൂട്ടാമിക് ആസിഡായിട്ട് കൺവേർട്ട് ചെയ്യാനും അലാനിനെ പൈറുവിക് ആസിഡ് കൺവേർട്ട് ചെയ്യാനും ഈ ഒറ്റൊരു ട്രാൻസ്ഫർ ഓഫ് അമിനോ ഗ്രൂപ്പ് കൊണ്ട് സാധിക്കും. വേറെ എക്സാമ്പിളാണ് ദിസ് ഈസ് അസ്പാർട്ടിക് ആസിഡ്. ഈ അസ്പാർട്ടിക് ആസിഡിന് ഈ അമിനോ ഗ്രൂപ്പിനെ എവിടത്തേക്ക് ട്രാൻസ്ഫർ ചെയ്യും? ഇവിടത്തേക്ക് ട്രാൻസ്ഫർ ചെയ്യും. ക്ലിയർ? So this is aspartic acid transferase. So this aspartic acid becomes oxaloacetate form ചെയ്യും. Then alpha ketoglutaric acid becomes glutamic acid converted. Okay, so simple process. These are the two examples under transamination.